Hello, Anna. Anna Wu.
Yes, Professor.
Did you ask him a question at the end of the last lecture?
Um,
It wasn't you.
yes. I asked about the boundary between positive and negative on the plot.
Yes. I want to add something. You
Uh,
know, although I explained to you that's you know very arbitrary, but you can always include a negative control there,
oh,
right? So you have
yeah.
you have an antibody that have the same color, like
Mhm.
it's a red fluorescence conjugated antibody, but that antibody is a non-specific antibody. I won't recognize anything on the cell surface. Then, you know, you should always have a negative in that dot plot.
Yeah.
So
Mhm.
you can use that to set the boundary.
Oh, I see that.
Okay, so that's one way to set the boundary. But as I said, still it is going to be arbitrary, as
Yeah.
you see from the slide. They they described that as lean negative and low.
Hmm.
Okay, so
Yeah.
it's always going to be arbitrary. So I thought about that. I just want to include that addition to
Thank
explain.
you, Professor. Yeah.
You're welcome. You're welcome. Okay, so we are going to start today's lecture. I hope we're going to have a relatively short one, since I always, push to the neck of every lecture. I feel bad, but I hope you kind of understand me. It is for advanced topics like this. It is very hard to talk just about 40 minutes. It feels like you didn't cover anything. And then once you describe several aspects of the topic, and then all of a sudden it's to the end 1 hour. So it's hard to manage, but hopefully in the future when I teach this 1 hour class, I'll be better. Okay, so today we're going to discuss mesenchymal stem cells and satellite cells. Cells related to uh, uh, skeletal muscle and skeleton. So we start with mesenchymal stem cells. That's another stem cell that you find within the bone marrow. The bone marrow indeed is a very wonderful source of stem cells. You have a osteoblast, which is not necessarily stem cells. They're probably just classified as a progenitor. It can give bone, and then you have hematopoietic stem cell that can give rise to all the blood cells in circulation. And now today, I'm going to tell you there is this second population of stem cell there, which is very abundant, called mesenchymal stem cell. So just like in previous occasions, so we start with history. So this scientist, Russian scientist, is named Alexander Friedenstein, who's the first to describe mesenchymal stem cells. So basically, in 1968, he found if you take the bone marrow and then you transplant this bone marrow to a rodent kidney. To be precise, you put the bone marrow under the kidney capsule. So what's going to happen is bone formation there. So under the capsule of the kidney, now you start to observe something hard. Bones are developed within. So what's going on? Apparently you would think maybe there's osteoblast there, right? Because osteoblast is progenitors that will give rise to new bone. But it turned out not to be the case, because osteoblast will be associated with the bone. It's on the surface of the bone. Therefore, when you do just the bone marrow isolation, you don't usually have that part, have the osteoblast in the preparation. What you get is the soft tissues, all the soft tissues. So he then tried to figure out what is going on. And then 2 years later, he found that if you will plate the bone marrow cells, now again is the whole bone marrow. If you collect the cells, you have some cells that will attach, some cells will not attach. So the cells that are easily attached to the culture surface are different from hematopoietic stem cells. That's what we have described previously in the previous lecture. So the hematopoietic cell will give rise to lineages within the hematopoietic system. And now you have additional cells, very abundant. They attach to the cell surface. And then he even found that those cells are clonogenic. So those cells can form individual clones from a single cell. Okay, and then he continued to work on that because he was trying to figure out what cells, were differentiated into bone under the kidney capsule. So he tried to replicate that observation in culture. He feel like if some cell will become bone under the capsule of the kidney, those cells should be able to differentiate toward bone tissues just in culture, in vitro. And then eventually he was successful. He found those clonogenic cells can give rise not only to the bone, but they can also give rise to chondrocytes and adipocytes. So chondrocytes is the cell for cartilage. Okay, adipocyte is the cell for fat, adipose. That's the name for another name for fat. And osteoblast apparently is the cell that will give rise to bone. So he was kind of amazed by that. You have some cell in there that can give rise to these 3 cell types once this cell will differentiate. And then he later on proved that it is this clonogenic cells. If you inject this, you transplant this underneath the capsule of kidney, they will form bone there. So later on he proved that it is this clonogenic cells that will give rise to bone in the kidney. So I don't think he named this mesenchymal stem cells, but apparently this is some kind of stem cell there. If you notice the timeline, his work is in 1968. That's after people started to describe hematopoietic stem cell in the bone marrow, and he, of course, raised this hypothesis or a theory that in bone marrow you have additional stem cells there that are not responsible for blood cell formation, but for other cell types, such as listed here, chondrocytes, adipocytes, and osteoblasts. So that's the origin of mesenchymal stem cells. But although, same amount of time has passed, since almost the same, since the discovery of hematopoietic stem cell and mesenchymal stem cell, somehow this field, the mesenchymal stem cell research field are not developed as well as the field of hematopoietic stem cell. Hematopoietic stem cell research, laid the foundation of almost all the adult stem cell research, but here somehow I think I can give you some reasons. This has become a very strange topic and strange research community. Later on, I'm going to describe to you that not only bone marrow, but a lot of other tissues can also give rise to this so-called mesenchymal stem cells. So it is very easy to get. Not like many other adult stem cells, you will have to take out the organ and then mince the organ and then use a very difficult approach to isolate those cells. But here, it is very easy. You can do a liposuction, you can just rinse them off and probably will get some mesenchymal stem cells into culture in no time. And since those cells are clonogenic, you can amplify them. You can get a lot in a month. So I would say this is the most widely studied. It is also, you know, explored by commercial companies. You know, they try to put this in therapy, into therapy use. So it is easily accessible and widely studied, and also perhaps the most used one in therapy attempts. So there you have a lot of noises accompanied the true experimental findings. You have a lot of things that's not true. People just publish and people just try to claim this works in a therapy setting, but once you start to do a lot to do careful work to prove that, it won't work. So nowadays, if you hear somebody said that they have a stem cell that can be used for therapy, you know, with attending this lecture, I hope in the future we'll know that they most likely are using mesenchymal stem cells. 95% of chance they're talking about the mesenchymal stem cells, and most likely the work is not carefully done. Most likely it won't work. So that's why I say it is a strange community, and it is a very, very strange topic. At one point, some people, in the research field, they hate this so much. They proposed in journals that we shouldn't call this mesenchymal stem cells. We should just say this mesenchymal stroma cells. You know, the name stroma means this is tissues that play supporting roles, such as blood vessels and connective tissue in an organ, and those are called the stroma of the organ. It's not the main functional cell, but connective tissue and blood vessels that provide the supporting role. Those are called stroma. So they feel mesenchymal stem cell, it is from the connective tissue and from the blood vessels. So they propose we should use mesenchymal stroma cells, which is also MSC, right? Instead of saying they are mesenchymal stem cells, because they feel it's very hard to find evidence to fulfill the criteria we described. That's self-renewal and differentiation. But I believe that's unfair, because later on, or maybe not even later on, at that time, scientists have already provided evidence that these cells do fulfill the criteria of stem cell. So let me explain the development in the field over time. So at the beginning, like Alexander did, basically you take the bone marrow, that's whole bone marrow. You probably just mince the bone marrow a little bit, and then you put that in under the capsule of kidney. Now bone formation. So the conclusion there would be, okay, bone marrow has osteogenic potential. So you put bone marrow there, you have bone. So bone marrow has osteogenic potential. That's the conclusion there. And then they try to find a subpopulation. What they did is they cultured the total bone marrow to a culture surface, but they only collected the adherent cells. Okay, any cell that are in suspension, most of the hematopoietic stem cell will be in suspension. Okay, so they only collect the adherent cell and then use those adherent cells to do the same experiment, again, bone formation. The conclusion here is that the osteogenic potential resides in the stroma. It's not the hematopoietic stem cell, it's cells in the stroma. And then they try to further get the cells into subpopulation or to more defined population. What they did is they culture the cell, make the cell to form colony. That's again done by the Russian scientist. He found that the stroma cell can form colonies. You notice that let me see if you can see my mouse. So the CFU means colony forming unit, but you have an F to follow CFU. It's a colony forming unit fibroblast. So basically, it's a colony forming unit, but it looks like fibroblast. If you have any experience in cell culture, you know if you have, for example, epithelial cells or cells round in shape, when they form a colony, the colony is going to be tight. It's a very tight colony. The cells are in close contact with each other. So those colonies are tight colonies. But if you have fibroblast, fibroblast is spindle-like. When you have a colony formed by fibroblast, it's not as tight. The colony is kind of loose. You know, you see cavities between the individual cells. So the morphology of those two types of colonies are different. That's why here they use a CFU-F to describe the colonies, you know, formed by fibroblast. But again here, what they show in this step is the cells, the clonogenic cells have osteogenic potential. Okay, so it's the clonogenic cells, non-hematopoietic cells, fibroblast-like cells in the marrow that have the potential to generate bone. That's what they found in that step. And then they have, in the next step, they isolated the single CFU-F. That's a single colony there. They isolated the single colony and put that under the capsule of kidney, again, bone formation. Now, the conclusion is even more precise. They say it's clonogenic stromal progenitors that are multipotent, that can give rise to bone and perhaps other tissues as well. And eventually, I think that's the most important step in stem cell research, to prove, you know, to prove it is indeed stem cells that is responsible for this differentiation in this animal experiment. So basically what they did is they isolated single cell. So from step 4, they get a single cell from that colony, and then they make that single cell to grow to form a colony. And now inject that colony to form a bone, and then do that again. Take that, you know, the injected cell under the kidney capsule and take that back, isolate a single cell out of it, and then again make the single cell to grow up as a single colony, and then inject again. This way they did multiple times. Now they are able to claim, okay, these cells can self-renew. They can, you know, give rise to themselves. And of course, the differentiation were proved at the very beginning. So the cells becomes more specified cell type. Now we have fulfilled the 2 criteria. So this is indeed a stem cell. Okay, this is stem cell. Mesenchymal stem cell is stem cell, but maybe we can say not everybody's mesenchymal stem cell is a stem cell. When people don't do careful work, maybe they just collect some cells without character those cells carefully. Maybe those are not stem cells. But in bone marrow, there is this cell type, MSC, that fulfills the criteria of stem cell. You know, to regulate the field, the research field, and this organization called the International Society for Cellular Therapy, ISCT, proposed this criteria to define MSC. So first, I hope you find this kind of strange. You know, usually it's not this type of organization that will propose criteria for something. So you're usually academic society. So from here, you can say, you know, this is indeed very heavily invested by people who are interested in cellular therapy. That's why I say, you know, that if somebody's talking about therapy, most likely they're talking about mesenchymal stem cell. So here we have 4 criteria. I'll explain one by one. First, the cells will have to be able to adhere to tissue culture plastic. So it can't be hematopoietic stem cell. Those usually won't attach to tissue culture plastic. So it's a very, very loose criterion there as the very first one. And then the second is expression of surface antigen CD markers, CD73, CD90, and CD105. Okay, so it's positive markers, must have those markers. And then the next criteria is the absence of markers. Other CD markers must be missing. Apparently, the CD markers that must be absent from the stem cell are markers that are associated with lineages, with different lineages. They're lineage markers. So these markers must be absent. Well, at the same time, you must have the CD73, CD90, and CD105. And then the fourth criterion is the ability to differentiate into three cell types, adipocyte, chondrocyte, and osteoblast, or osteocytes. So basically, mesenchymal stem cells, as was described at the very beginning, must be able to differentiate into three cell types that belongs to three tissues: fat, cartilage, and bone. So even with these four criteria, it's still very easy to fulfill. A lot of cells, cells from quite a few different places can do that, can fulfill that four criteria. So on this slide, I just give you a very typical study that describes the tri-lineage differentiation of mesenchymal stem cell. So you put the cells in culture, you give them the right condition, sometimes including some growth factors, and now in individual culture vessels, with the correct culture condition, you demonstrate these cells can. It doesn't have to differentiate toward these three cell types at the same time, as long as these cells can differentiate to one of these three at a time, but you need to do all the three experiments to show that the cells can differentiate into three lineages, three cell types. So I just give you some the image as well as the method. To demonstrate fat differentiation, the reagent usually used is called oil red O. The oil red O is specific for lipid droplets. Lipid droplets is abundant in fat, in adipose. So with this oil red O, it's just a chemical. And you can demonstrate, okay, now you have fat differentiation. So in the middle, that is a study of cartilage differentiation. The chemical used for staining here is called toluidine blue. Toluidine blue is specific for cartilage differentiation. And finally, what's shown there is bone differentiation. Bone can always be stained with a substrate to demonstrate bone differentiation. The substrate is specific for alkaline phosphatase because in bone, alkaline phosphatase is abundant, and now with the proper substrate, you can generate color there. Okay, so although this is positive staining for alkaline phosphatase, actually what you are applying is a specific substrate for alkaline phosphatase. So this is how you demonstrate the tri-lineage differentiation from mesenchymal stem cells. And here, it just illustrate to you, where you can identify mesenchymal stem cells. So although I listed those tissues, that's what was listed from our textbook. The original place will be bone marrow. You can get it from blood, from circulation. You will get mesenchymal stem cells. And amniotic fluid, that's what's present in early life. The fluid protected the fetus giving him or her a very good environment, a very comfortable environment. So if you collected the fluid, you centrifuge down, you will find the mesenchymal stem cells there. Umbilical cord. Within the umbilical cord, you can collect hematopoietic stem cells in umbilical cord. You can collect the hematopoietic stem cells there, but you can also collect mesenchymal stem cell. So that organ, umbilical cord, is kind of like a bone marrow. A placenta as well, dermis, adipose tissue, and teeth. So that's why, if in the future your wife or yourself or somebody from the family will give a birth, and then usually the doctor may ask, do you want to collect the umbilical cord blood, put into a bank, a tissue bank, or do you want to collect the placenta? The purpose is to keep a tissue bank of stem cells, maybe hematopoietic stem cell, apparently will be found there, as well as mesenchymal stem cell. So if something will happen in the future, they can store the cells and then start to prepare mesenchymal stem cells, hopefully for some therapy use. But very rarely, we have demonstrated the usage of mesenchymal stem cells. There is one approved drug, I believe in Australia. I can't remember the country, but there is a drug that's already approved. That drug is a preparation of mesenchymal stem cells. So hopefully I have a chance to describe that drug. That's the only drug originated from mesenchymal stem cells. So that's the only one. And then when people do liposuction, that's an option. Usually they get offered, will you like us to prepare mesenchymal stem cells for you, or freeze down some mesenchymal stem cells for the use in the future? So what you can see here is it can be isolated from many places. You started to wonder, are they the same cell? If you remember the 4 criteria there, the 4 criteria are not that difficult to fulfill. Attachment to plastic surface. A lot of cells will do that. And some cell surface markers, not hard to fulfill. And then apparently they have a lot of things in common, right? They have a lot of things in common, but from different places, are they different? That's a topic that scientists would like to explore. That is important. And here I show you one example of that of work along that direction. So the work was done by Michael Lineker and Irving Westman at Stanford. Now again, Irving Westman. So he felt, these 2 scientists feel like they want to do something. As I said, this is a messed up field. People don't even like to call them mesenchymal stem cells. So they try to figure out whether stem cells from different places are actually different. Are their potency affected by the anatomic place where they were isolated? So they did work with long bone, and they isolated mesenchymal stem cells, but they further purified the cell. They didn't go with the criteria proposed by that organization, but then what they used is surface markers, like an integrin, the alpha V. It's an integrin there. They also used the CD105 and the CD200. The CD105 was one of the markers proposed by that international organization. CD200, I believe, is not one of the markers. So CD200 is, let me go back to see that. No, it's not listed there. That's a new marker that they have identified. So what they defined is the stem cell bear the marker alpha integrin, but does not have the marker CD105 and CD200. They used that integrin to define a stem cell population, but they did both positive and negative selection, as you can see there. So these cells, if you go with the criteria there, according to the criteria, mesenchymal stem cells should be CD105 positive, but here, Westman and Lineker defined a mesenchymal stem cell population that don't express CD105. Okay, CD105 is used as a negative selection here. See, they used the integrin to define their stem cells. And then they found these cells can self-renew and can differentiate. They called these cells skeletal stem cells. Skeletal stem cells, because these cells can give rise to bone and cartilage. So they used additional markers to define the differentiation process. So when CD200 becomes positive, and they call these cells pre-BCSP. So BCSP is the BCSP is the hematopoietic supportive stroma. This is a long name. It's a bone, cartilage, and I don't know how they get that BCSP. Supporting stroma, I couldn't figure out, but they call it BCSP. Okay, that's the name they give to these cells. It's a multipotent cell population that eventually can give rise to cartilage, bone, and stroma. The stroma means supporting cells in the bone marrow. Okay, they're not. I don't know whether these are mesenchymal stem cells. I mean, just the stroma cells are just the cells present in the marrow that play a supportive role. So here, the strange thing, maybe you have noticed, so these cells are multipotent. They can give rise to cartilage, bone, and stroma, but you don't see fat, right? You don't see fat, and you don't see what else? According to that, you should have chondrocyte. Chondrocyte is here. You have cartilage, right? Cartilage is chondrocyte, and then you have bone. That's osteoblast. Okay, so what you don't have here is fat. So these cells do not give rise to fat. It can give rise to cartilage and bone, but not fat. So if you compare this to the differentiation potential of mesenchymal stem cells, you feel like, well, these cells may be a more specified stem cell than mesenchymal stem cells. But the tissue origin that they started are the same. It's marrow, although here is long bone marrow. Whether these are two different identity, right? I don't think so. Maybe the so-called skeletal stem cells is a subpopulation of the stems of the general mesenchymal stem cells. So I guess this paper is important because the conclusion here is that maybe mesenchymal stem cells share common characteristics, but at different places, they have different potency because maybe their environment will change their different potency, or maybe they're different epigenetically. Somehow they're different. Therefore, maybe in the long bone, they prefer to get differentiated towards skeletal progenitors. Therefore, eventually become cartilage and bone. Well, in adipose tissues, you isolate the mesenchymal stem cell. Maybe those cells will like to become naturally. They would like to become adipose. So that's what they concluded here. Okay, so as you can see, this is just some of the rare attempts in this field to further classify the mesenchymal stem cells to finer subpopulations. It's just one example, but that conclusion may not be true. It's just the start of further exploration of subpopulation of mesenchymal stem cells. So this group of scientists did another piece of work to show that the matrix or the supporting material for the mesenchymal stem cells play a major role there. So in this experiment, what they did is just a mesenchymal stem cell collection, and then they put mesenchymal stem cells onto different substrate. So in this different substrate, they use cross-linking and the concentrations of collagen to change the hardness of the substrate. So they use this, the KPA to describe how hard, how stiff that material, that substrate is. So for reference, in the brain, the stiffness is usually 1 kPa, 1 kPa. In muscle, it is 10 kPa. And then in bone, it's collagenous bone, it is 100 kPa. So it's easy to understand. Bone is very hard, muscle is softer, the brain is super soft. Okay, the muscle is compared to brain is still hard. So you use this to describe the stiffness in different organs, and in cell culture, they try to simulate that by cross-linking collagen to the matrix. They can change, the stiffness of the culture matrix. So what they have found is if you apply a low stiffness, which is 0.1 to 1 kPa, and now the mesenchymal stem cells can even differentiate toward beta-3 tubulin positive cells. Beta-3 tubulin is a marker for neurons. So the mesenchymal stem cells can even differentiate into neurons if the substrate, has the stiffness of the brain. And then they describe if you increase the stiffness to 8 to 17 kPa, you can generate muscles. So MyoD is a marker for muscle, and now you can generate muscles. And then if you further increase that stiffness, eventually you can make bone. So CBF alpha 1 is a marker for bone. So their conclusion here is maybe the environment is very important to determine the differentiation potential of mesenchymal stem cells. So one more thing you learned here is it seems that mesenchymal stem cells have additional potency. They will not only differentiate into that classic the classic three lineages, the tri-lineages, which is chondrocyte, adipocyte, and osteoblast. Those, usually is the gold standard for the differentiation potential of mesenchymal stem cells. But here, it looks like if you change the substrate stiffness, you can make them to differentiate into different tissues, even beyond the three original described. So what's the conclusion here when you compare to the study shown on the previous slide? I guess, well, maybe the anatomic place play a role, but the environment also plays a role. I guess because that field is a very strange field. The work hasn't been carefully done for the past few years. And maybe now we know that you can get clonogenic cells, and you can also further subdivide the cells to finer subpopulations. Maybe we'll learn more about mesenchymal stem cells. But actually, we should learn more because there's a reason, why commercial entities try to use that for gene therapy purposes. There's a reason, it's easy to accept, easy to get to the cells, and then everybody can generate a bank of your mesenchymal stem cell. So you don't have to worry about immune response. It's always abundant. You can amplify the cell, it is a good source to be fair. It is a very good source. We just need to do more careful work so that we can define that better, so that we learn how to use them in therapy. So I'm going to use 3 or 4 slides to describe the last this stem cell population I want to describe. So this is called satellite cells. Satellite cells is muscle stem cell. Someone proposed to use this MUSC instead of satellite cells because satellite cells won't give you the straightforward impression what the cells are. Satellite cell, okay? They call this muscle stem cell, but there's the same thing. There are adult stem cell that's within skeletal muscles. So there are the abundance of them is there are about 2% to 7% to 7% of all nuclei in the myofiber that are satellite cells. But first of all, you need to understand that the muscle is multi-nucleate cell. Some muscles can have 100 or more nuclei. So how things work is these satellite cells will fuse. They get activated. They will start to replicate, and then they get fused together to form new muscles. So that's why the muscles, no matter new or old, they always have a lot of nuclei. It's because satellite cells are a single nucleus. But when they fuse, many of them fuse together, now you have myofibers, but in myofiber you have a lot of nuclei. As shown here, the blue colored are just nuclei, a lot of nuclei there, right? But let's look at A here. You have a lot of nuclei, but you do have some cell around the edge of the muscle fiber. It's right under that basal lamina. Under the basal lamina, you will find satellite cell. They are always around the edge of the muscle, under the basal lamina. Okay, so their function is to regenerate. So our muscle always regenerate. Sometimes it's because of a disease. Sometimes it's because of you exercise. You go to the gym. You train yourself. Essentially what will happen is you're going to damage your muscles. The muscle will get cleared. It's damaged, and now satellite cell will make a bigger, even more powerful, more muscles in the place where you have damage. That's the mechanism how you generate muscles. Damage and then repairing or regeneration by satellite cells. So this scientist, Alex Morrow, was the one who first described that at about the same time as the discovery of hematopoietic stem cell around 1961, 62. But that scientist didn't, the scientist didn't show the function. He observed those satellite cells because others demonstrated the function of hematopoietic stem cell. He said maybe these cells perform the same function in the muscle, but he didn't demonstrate that. But he's still credited to be the one who found the muscle stem cell. So here I'm going to tell you an animal model that's called a muscular dystrophy animal model that is used for in vivo demonstration of stem cell potential. So this is an animal model of a disease called muscular dystrophy. So this dystrophy is a disease because of mutation on the dystrophin gene. The dystrophin gene mutation will cause this muscular dystrophy. And then people, developed the animal model to simulate that disease. Although this animal model is a little milder than the real symptom observed in the real disease in humans, but still, it is used as a model for that disease and also for the study of regeneration. So basically, in this MDX animal model, there is also always damage because there is a mutation in one of the key genes of muscle. It's a dystrophin, it's a structural protein of the muscle. When you have a mutation there, it always causes damage in the muscle, but at the same time, you always has regeneration there. So the satellite cell will constantly repair the damage. So because of that, now you can inject, you can transplant a single muscle fiber, Remember, it's just a single muscle fiber is a single muscle cell, although it's quite big, but it's just one cell. Okay, you can put a single muscle fiber that you already have about 7 or so satellite cells there. You can put a single muscle fiber there and observe how that single muscle fiber will contribute to the regeneration within that recipient animal. So as you can see here, you can use some genetic marker to mark the stem cells, the transplanted stem cell. At the beginning, you have 7 positive, this beta-galactosidase positive cells, but if you put it into this animal, you see how many. It will generate a lot of cells. That means the cells can self-renew and contribute to the regeneration process. So that's why this model becomes a popular model for that purpose. So here, it's just to show you, this is from the donor, because only the donor will have this genetic mark there. The donor have a positivity for this beta-gal, and also this donor cell also is positive for PAX7. PAX7 is a transcription factor specific for satellite cells. So when it is positive, it means it is a satellite cell. Okay, so that illustration just showed you the donor cell is a satellite cell. Prove it is a satellite cell. Okay, so how's the cell defined? How's the cell defined? Previously, when we talk about mesenchymal stem cell, hematopoietic stem cell, we use surface markers, right? CD markers, flow cytometry. But here, things are a little different. Since the starting material for hematopoietic stem cell and mesenchymal stem cell are marrow or adipose, you can easily make a single cell suspension. You can have a lot of cells. You can do flow cytometry when you have a lot of cells. But here, well, the source, the tissue source is limited. And also the number of satellite cells will be much smaller than that. So instead of using flow cytometry, people use genetic method to track the cells, like a Creed-LOXP system, which we have described. Okay, so here I just want to explain to you the presence of those markers during differentiation of satellite cell. So satellite cell will have to have expression of PAX7, which is a transcription factor. But also, the quiescent satellite cells have an active MEF5 locus. Pay attention to this description. So there's no evidence that satellite cell will express MEF5. The quiescent satellite cells will not express MEF5 protein, but the genetic locus somehow is active. That means if you put a green fluorescence protein into the genetic locus, you're going to see green fluorescence protein. However, the MEF5 protein itself, the protein itself, is not manufactured. Okay, so in other words, maybe the gene MEF5 will be somehow present, but you don't have protein in quiescent satellite cells. But later on, when the satellite cells get activated, when they start to self-renew, right? When they start to replicate, you will start to have MEF5 expression. Okay, the MEF5 is slightly later than the expression of PAX7. And then another protein, transcription factor, MAO-D will be expressed. I hope you still remember when we discussed pluripotent stem cell reprogramming, we have MAO-D as the major discovery by Weintraub. Weintraub showed that the MAO-D is differentially expressed between muscle cells and fibroblasts. So he used the MAO-D, he put MAO-D into fibroblasts and converted fibroblasts toward muscle cells. Here, later on, people did careful work. They know when exactly MALD will be expressed. It is when the quiescent satellite cells get activated and they almost completed self-renewal. And at that time, MALD will start to express and then start to give the cell the identity of muscles. And later on, when the cell become committed muscle progenitors, that means they now will become muscle cells. This cell will start to express myogenin and MRF4. So these are several transcription factors that are expressed during the course of muscle satellite cell differentiation. The critical thing is you must know that PAX7 is the transcription factor specific for quiescent. I'm sorry, not only for quiescent. As you can see here, the PAX7 expression is the highest in quiescent cells, but it persists for quite some time. So you can only rely on the appearance of other markers to describe the course. So PAX7 is always expressed. Okay, so pay attention to MEF5 as well. The locus is active in quiescent cells, but the protein is absent. Okay, lastly, this is the niche of satellite cell. To make it extremely simple, in the niche you have supporting the basal lamina, the support that provide the support, and also the other cells such as the blood vessel associated cells will provide angiopoietin and tie to the PAX7 positive satellite cells, and that the satellite cells will be anchored to the basal lamina through laminin and integrin. So that's just some structural connection. The important signal transduction here is through notch signaling and Wnt signaling. To make it simple, Wnt will cause the cell to start to differentiate, while notch will keep the cell in a quiescent state. So those two coordinate with each other. So the notch ligand, the notch ligand is delta. Delta is from the muscle fiber, and on the satellite cell you have notch receptor. Therefore, this pair will keep the cells in quiescent state. But upon activation, the canonical Wnt signaling will increase, antagonizing the effect of notch. Therefore, the cells will start to proliferate and get on the road of differentiation. So notch and Wnt, the balance between them decide whether the cells will be in quiescence or the cells will become activated and start to differentiate. So that's the notch signaling is interaction between the satellite cell and the muscle fiber itself. Okay, that's end with. Oh no, we still have one more very important slides here. So what's shown here is based on the observation that we lose satellite cells over time. So at birth we have about 30%. No, it's mouse. In the mouse about 30% of the nuclei is satellite cells. That's a lot, right? But in adulthood, that number dropped to 4% of all the nuclei. But in aged individuals, that number dropped to 2%. So why you see a drop of the cells of stem cells? So at the beginning, people feel like maybe over time you have a more mutation within the stem cell population, or more oxidation within the population. Eventually, you just lost the cell. They get damaged, they get cleared, so you don't have the same amount of cell because of that. But by this experiment, it turned out that is not true. So this experiment is called parabiosis. Parabiosis. So this is called parabiotic mice. So what happened here is they open the flaps of two mouths and they get them linked together. So they link these two animals for a long period of time. So the circulation of these two animals will be the same circulation. Okay, it's the same blood that's circulating in these two animals. Of course, these two animals must be compatible. So you can do this experiment called parabiosis. What's shown here is if you pair two young animals, you create an injury using dry ice on the muscle, you're going to have regeneration, perfect regeneration. If you pair young and old animals, you do an injury on the old animal side, still you can have good regeneration. But if you pair two old animals, now you do a injury, there's no regeneration. So you see that injury is always on the right side animal. But if you link that old animal with the young animal, somehow the regeneration can get restored. That means in circulation, there is some factor that goes to the stem cell niche, activate the muscle stem cells for that old animal. Then you can't say it is because of cell loss, but rather that's the environment. So the old animal doesn't have a good environment as a young animal. So the niche is different. That's what this study proved. So what is the critical factor? I guess we're still learning about the critical factors there. You know, based on the signal transduction, wind and notch plays a role, but maybe other factors will modify that main principle there, therefore contributing to the maintenance of stem cells within the niche. Okay, that's the end of today's lecture. I'm sorry again, push to the end of time. If you have a question, you can ask me. Otherwise, please feel free to leave.
Oh, Professor, hi, Evan.
Yes.
Yeah. So in another class, I kind of learned about